Hello everyone, my name is Roy from Eureka Bio. Today I'm going to give you a presentation on lentiviral vector production based on the stable producer cell lines. Here is the disclaimer from Eureka Bio. As everyone watching this video knows, we are in the era of gene and cell therapy. With more and more clinical trials in this field, the viral vectors are the key to the cell and gene therapy. With the increasing demand of these therapies, the viral vector manufacturing is becoming a bottleneck of this industry. So let's take a look at what challenges we are now facing. In 2019, the National Institute for Innovation in Manufacturing Biopharmaceuticals has published the Gene Therapy Roadmap, explaining the current manufacturing challenges and technology development. This can be summarized in 4 categories, which is plasmid production, packaging cell line, upstream and downstream challenges in the viral vector production. In today's presentation, we are honored to introduce our answer to the challenges that I have just mentioned. We called it the EuLV system. It is an inducible lentiviral vector stable producer cell line with scalable process technologies. Generally speaking, the EuLV system is an inducible stable producer cell line, adapted to high cell density suspension culture in the chemical-defined medium. This stable producer cell line no longer requires transient plasmid transfection. Instead, all the required packaging genes such as VSVG, REV, GAG-PRO, as well as the customized therapeutic genes, or commonly called gene of interest (GOI), are stably inserted into the genome of the producer cell lines. And the production of lentivirus is simply achieved by chemical induction. This slide shows the schematic diagram of the EuLV system development and the lentivirus production. It mainly involves 3 phases. In phase 1, all the lentiviral packaging genes, VSVG, GAG-PRO, REV, and the components of the molecular switchers are stably inserted into the genome of 293 T cells to construct the EuLV packaging cells. And the monoclonal cell lines are screened and tested. In phase 2, the customized GOI is doubly inserted into the genome of the best clone of the EuLV packaging cells to construct the producer cells, and the monoclonal cells are screened and tested. In phase 3, the EuLV producer cells are cultured on a large scale in a feed-batch manner, and the lentiviral vectors are produced when the inducers is added. So here comes the question, how to pick up a superclone? There are 3 key factors. First, higher population mean. Second, higher population variance. And third, large number of screening sample size. In these figures, x-axis is the viral population titer, when y-axis is the numbers of the clone. We have a mixture of the cell lines. By our technology in cell line construction, we increase both the average production titer and the performance variety in the viral vector production. This will guarantee a good starting point for the clone selection. Also, we adopted an automation-assisted high-throughput screening platform, we called it EuBioX, to ensure that we will not miss the few super clones. To increase the scale and accuracy of the screening, we also built our own automation facility, EuBioX, as we can see in this slide. It could perform routine operation of the cell culture, monoclonal cell screening, cell-based and it has adapted to 384 to 6 well plates. With this equipment, we can screen over 10,000 individual cell clones for each round of the experiments. And also, we have also built an EuBioX non-staining live cell analysis system. This device is part of the automation system shown in the previous slide and can take photos of 384 well plate in less than 5 minutes. Using this device and our algorithm, the device can record, trace, analysis, and pick the monoclonal with best performance. This slide is very important. It shows the main data to construct the EuLV system, including the data for packaging cell line screening, evaluation of the good clones, and the producer cell line screening. During the development of the packaging cell line, the human PGK luciferase RES EGFP was used as the gene of interest. It is about 3.5 kb size. Transduction titer quantitation of this lentivirus were performed in HT1080 cells, either by luciferase assay or by flow cytometry evaluating EGFP positive cells. These 4 figures at the top illustrate the screening results in the construction of the EuLV packaging cell line, which include 1 monoclonal screening step to identify single clones with good growth curve, and 4 functional screening steps, which identify the best production clones by measuring luciferase transduction titer after transient transfection of the luciferase plasmid. The x-axis was the log scale of the luciferase unit in HT1080 after transduction, and the y-axis indicate the number of the clones. The red line is the positive control, which represents the transduction titer of the lentivirus produced by the transient transfection 293 T cells with 4 plasmid. And the dashed line was the average titer of all the clones in that screening step. So after 5 rounds of screening, the average transduction titer was 10 times higher than the positive control, the red line. After these experiments, we got few clones, which I've shown to be the best performance. Then we want to see if they are truly optimized. In principle, if all the packaging genes in the packaging cell lines are fully optimized, for example, the copy number, the expression level, and the relative ratio, no increase of the titer should be observed after further transfection of any packaging plasmid. As shown in the bottom left, the data indicate that after transfection of any packaging plasmid or any combination of this packaging plasmid, no significant increase in the viral titer was observed in the first 3 clones, which means the packaging genes in these packaging cell lines are optimized. The 4th is the control. Finally, see the bottom right figure. We choose one of the packaging cell line to construct the producer cell line. This figure shows the titer results during the 4 rounds of screening. After 4 rounds screening like this, the average titer in the screening 4 increased nearly 100-folds compared to the average titer in the screening 1. One of the best producer clone was selected for the further process development. The first step of the process development, the cells need to be adapted to the suspension culture, and the culture medium selection is the key. We have screened and tested few commercially available medium. All of them are chemically defined mediums. Figure 1 and 2 at the left shows that the CDM2 it has the highest cell culture density, which can exceed 10 million cells per mil, and maintain high cell viability after 6 days of continuous culture. More importantly, this medium CDM2 did not interfere virus production, which you can see at the figure at the right. Secondly, we optimized the inducer concentration and enhancer formulation. We use the DOE method. We screen 4 compounds that can be used as enhancers. And after optimization, virus yield was increased by 80%. And this slide shows some data from our feed optimization study. In this study, we have achieved 2 goals. First is a feed batch process without medium exchange. And second is a suspension culture in high cell density, which is over 10 million cells per mil. With this feed, the virus titer was increased by 4 times. Medium exchange is not required, and this feed batch process was stable when tested from 5 ml to 300 ml in the shaking flask. Here, I want to emphasize on some crucial updates of the EuLV system. It is achieved by combination of few key factors, including the optimization of the cell line construction method, the cell screening method, and the redesign of the lenti genome transcription cassette, virus production process, and so on. The 3 figures at the left show the titer results for top 4 experiment designs A, B, C, and D during 3 rounds of screening. The x-axis is the transduction titer measured in HT1080 for each round of screening, and each dot here represents a transduction titer result from a single clone. After several rounds of screening, from 384 well plates to a suspension culture, clones from design B, design C, and design D all have the titer higher than 100 milliliter per mil, and the 03 and 18 clone from design B and 01B1 clone from design D was the best clone with titer higher than 400 milliliter per mil. Then, 3 top clones from each of the 4 designs were cultured continuously for 30 passages for the stability test, which lasted 90 days. The luciferase titer was measured every 3 passages, and data was summarized in the left. No significant titer reduction was observed for all the 12 clones tested. Clones in design D were slightly better than the clones from the other designs, with higher average virus yield and lower batch-to-batch variance. As seen in previous slide, we use luciferase assay for the virus titer measurement during the construction of the EuLV system. We also use the qPCR-based titer method in the producer cell line screening, and it is a universal method which is independent to the genome of interest. The first figure shows here the data from the producer cell line screening. The qPCR titer method correlates very well with the luciferase titer method, and the qPCR method is therefore a good enough method for the monoclonal screening. Another advantage of the EuLV system is that we can actually combine the virus physical particle titer P24 with functional titer together during the producer cell line screening to screen cell clones with high specific transduction activity. In other words, is with lower empty capsid ratio. The figure at the right, the x-axis is the transduction unit, and the y-axis is a P24 reading. It is an example here that the 2 clones in the blue circle produce less P24 but will have higher transduction titer. Another concern about the lentiviral stable producer cell line is the non-induction leakage. So we choose a clone with titer higher than 400 million per mil for the leakage study. The orange bar indicates the virus titer after induction, and the viral titer is over 400 million TU per mil. The background noise measured in 293T and the EuLV packaging cell lines were around 700 TU per mil, showed as light blue and light yellow. When cultured in the same condition as the inducer sample, the producer cell line, while without inducer, produced virus with the same titer as the background, around 100 TU per mil. Even the culture medium is concentrated for 100 times by ultracentrifugation, the viral titer measured is still indistinguishable to the background control, which indicates the virus leakage in the EuLV system was probably less than 10 TU per mil. Here shows the process data using the EuLV system in the wave bioreactor for 1 liter. The left diagram shows the main steps. Day 1, we saw the cell, culture in the flask, expanded to day 7. After transfer to the wave bioreactor, culture for another 5 days, when the cell density can reach 10 million cells per mil, then we will add the inducer and the feed to start produce the lentivirus. After 48 hours, which is on the 14th day, the virus was harvested, purified, and stocked. The two figures at the right show the cell density and the viability of the producer cells in a 1 liter wave bioreactor. Inducers are added on day 5 when the cell reach 10 million cells per mil. Cell viability remained close to 90% when the virus was harvested. Compared with conventional transient transduction, this production method using EuLV system has less cell debris due to the high cell viability upon harvest. After 5 steps of purification, the virus purity can reach 98% measured by HPLC. The table at the bottom shows the results after the purification. Around 5.3 to the power of 11 TU per liter viral yield in the culture medium, and 1.2 to the power of 11 TU per liter after purification. The lentiviral particles is over 97% purity, and the inducer is not detected. Here shows the 25-liter lentiviral production in the wave bioreactor. The process is not much different from the 1 liter scale process. The producer cell will transfer to 2-liter wave bag on day 7. Instead of induced for viral production, we expand the cells to 5-liter on day 10 and expand the cells to 25-liter on day 13. The induction is on day 18, and virus was harvested after 48 hours as previous. The total process is 20 days from cell thaw. The two figures at the right shows the cell density and the viability data when the producer cell are amplified and induced for the lentivirus production in the wave bioreactor. On day 10 and 13, when the cell density reached around 5 million per mil, cells were transferred to the next size of the wave bag. And on the day 18, inducer was added for the virus production. Same as the 1 liter data, cell viability is controlled over 80% upon harvest. And for the virus titer, we detected 3.8 to the power of 8 TU per mil in the culture medium, and 9.5 to the power of 12 TU before harvest, and 3.4 to the power of 12 after purification. We also constructed EuLV anti-CD19 CAR IRES EGFP producer cell line. This slide summarizes the monoclonal screening data, the leakage analysis data, and the enhancer optimization data. Here is the map of the GOI sequence at the top of this slide. It has the typical anti-CD19 CAR sequence. Additionally, it has an IRES EGFP sequence. So in total, it is almost twice the length of the original anti-CD19 CAR sequence. Then let's look at the figure at the left up. This is the monoclonal screening results. In the figure, the x-axis represents the viral titer measured by fax. And this will represent the second and third screening results. Each dot here represents a transduction titer result from one single clone. After 3 rounds of screening, the best clone can reach to 50 million TU per mil measured by fax. And let's see the figure at the right. It is our enhancer optimization. It increased the titer by 2.4 times. We found that for the different producer cell line with different GOI, they will respond slightly different to the enhancers. So it is very worthy to re-optimize the use of the enhancer for different projects. So let's see the figures at the bottom. It is for the leakage test. Let's see the one at the left. The packaging cell line without GOI worked as the negative control. Shown in this figure is the white bar. And the blue bar shows the virus titer after induction. And the orange bar shows the titer of the non-induced sample, which is after 100 times concentration by ultracentrifugation. Here, the dot plot of the fax results. The first up is the negative control, and the second, the induced figure, is the induced titer with only 0.78 microliter virus. And the third one, the not induced one, the result is after the addition of 100 microliter of 100 times concentrated sample. The same as previous data, without inducer, no virus could be detected in this culture medium for this cell line. Here we have the preliminary process data of our EuLV anti-CD19 CARIES EGFP producer cell line. The 2 figures at the top is the cell density and cell viability data in 1 liter wave bioreactor. Similarly, they are induced on day 5 and harvested after 48 hours. The bottom shows 1 batch downstream process data. Here is the purity data in the process step 1, 3, 4, and 6, measured by HPLC. And the purity, the transduction titer, and the recovery data were summarized in this table. And for this batch of experiments, we got 8.13 to the power of 10 TU per liter in the culture medium, and 2.91 to the power of 10 TU per liter after purification. From the result of HPLC analysis, the viral particle purity is 84.5%. To our experience, there is still a lot of room for optimization of this process, and we are keep on working on them. So after talking so much, let's look back on the key manufacturing challenges in the lentiviral vector production. By using EuLV, the stable cell line to produce lentivirus method, the deleted items means that EuLV has skipped over these challenges, when the ticks means that challenges has been solved by the EuLV system. By using the EuLV system, the process for lentiviral vector is significantly simplified. The EuLV system saves all the plasmid-related costs. It no longer needs the plasmid GMP. It reduced the size of GMP infrastructure. It reduced the manpower and operation costs to manufacture the lentivirus. It simplified the QC process by skipping over the plasmid parts. The EuLV system not only largely improves the production efficiency, but also bring your lentivirus vector to a stable, high quality, reproducible, and easy to scale up manufacturing processes. So here I would like to have a really quick recap of the EuLV system. Comparing with conventional plasmid transfection method, our EuLV system uses stable cell line and the inducer to produce the lentiviruses. It adapted to the suspension culture using chemical-defined medium and a feed batch production processes. Lentivirus production using EuLV system is simple, with much less batch-to-batch variance. After a systematic and comprehensive evaluation during the producer cell line screening, the lentivirus produced in the EuLV producer cell lines are more homogenized with significant lower empty capsid ratio. The titer in the culture medium can reach 800 million TU per mil, with at least 10 times higher than the traditional 4-plasmid transient transfection method. Importantly, it greatly reduced the production cost, which is crucial for the commercialization of the products. Currently, we start to provide the CRO service for the EuLV system. Client can just simply provide your gene of interest and the monoclonal cells can be delivered in 4 months. We also provide optional service such as GOI optimization, small-scale production, process development, and so on. And we have few projects already started in our facility. So last is a quick intro of our company. We are a biotech startup funded in 2014. We focus on the upstream of the GCT industry. Besides EuLV system, we also develop some devices for manufacturing cell therapy products. This is my presentation. Thank you.